So today I'm interested in talking about polygenic adaptation and how it can get concentrated in inversions. But my lab works in a variety of different study systems. We're really interested in evolutionary processes and adaptation to environments. And I'm particularly interested in the processes that generate and maintain diversity within species and how we might leverage adaptation through the past to predict how species will respond to climate change in the future. So I've worked in some terrestrial species, but also we have ongoing projects in marine fish and marine invertebrates and seagrass right now. But today I want to talk about polygenic architectures in the context of animal migrations. So animal migrations have long fascinated biologists and people. We have the Arctic tern, which chases a perpetual summer from the Arctic to the Antarctic and back every year. We have tiny insects like monarch butterflies that fly thousands of miles to overwintering grounds, to huge, enormous animals like humpback whales that travel halfway across the world's oceans in search of following food and migration patterns. So a lot of research has come to understand the reasons that animals migrate and sort of the reasons that they evolve these behaviors. And it has a lot to do with trade-offs between breeding and survival, or potentially chasing food. So on the right, for example, the wildebeest in Africa basically follow the rainfall that stimulates the growth of the grass that they like to eat, and they'll travel thousands of miles chasing this food source. But we know much less about how migration distance, the actual act of movement, evolves at the genetic and phenotypic levels. So for example, the genetics that have been done can distinguish, they've discovered genes that distinguish fish, for example, that migrate at different times of the year. They've discovered gene that distinguishes birds that go to different overwintering grounds. But all of these individuals in these populations are basically migrating, they're basically moving. And so these kinds of comparisons don't really give us insight into how the act of migration itself is evolving. And so to really get at this question of like, how does migration evolve at the genetic and phenotypic levels, we need a study system in which there's a lot of mixture, like migration between different ecotypes, but some individuals are moving long distances and some individuals tend to be more resident and not move as far. And so to understand how migration evolves sort of at like an initial level, we need migratory and non-migratory ecotypes within a species. An Atlantic cod is a marine fish that offers an interesting opportunity to study this, and multiple places in the northern Atlantic. For example, in Norway, Iceland, and Greenland, there are different substocks of cod that have different migratory patterns. So there's coastal cod, which tend to stay within fjord systems. And then there's migratory cod that travel hundreds of kilometers up and down the coast every year and are typically caught in deeper water offshore. In the Gulf of Maine, where my research institute is located, we only have a coastal ecotype. There's fish with different propensities for movement, but we don't have any stocks that undergo these really long distance migrations. So today, I'd like to look at 3 different questions related to the evolution of genetic architecture and migration. First, what are the phenotypic and genetic differences between these ecotypes that we see in nature? As I will explain, we'll see multiple large supergenes involved in this ecotype divergence. And so I'll dig into some of the modeling that my lab has done to try to understand why this evolves. And then finally ask, go back to the data and ask, okay, do the empirical patterns that we see in the system sort of support the theoretical predictions? And so the data set that we're working with was collected by my graduate student, Sarah Schall, who collected almost 80 fish offshore at 2 different locations in Iceland. These offshore fish are those putatively migratory fish. Another 80 individuals caught near shore in Iceland, which are the putatively coastal ecotype. And then another 100 or so fish caught near shore in the Gulf of Maine. And each individual fish we sequenced with whole genome Illumina sequencing to 15x coverage. And I just want to point out that this data set would not have been possible with all of our collaborators in the Gulf of Maine and Iceland who helped us collect these samples, and our collaborators on the bioinformatics, because this was an enormous amount of data. And the adaptive trait that we're interested in is drag reduction. And so a lot of biophysics research has shown that small differences in the shape of aquatic animals can have big effects on the drag that those animals experience in the water. And this research is used in the design of boat hulls and aircraft. And so we hypothesized that offshore fish will have more streamlined body shapes for long distance migrations. And so what we did was morphometrics on each fish, where we have these landmarks and we calculate the distance between the landmarks. And some of these distances are highly correlated with each other. And so they're colored according to the groups of traits that are highly correlated. And so we would only pick one representative trait from that group for our analysis. Does that make sense? Yeah. So for this first question, how do migratory traits evolve at the phenotypic level? Here are some representative fish pictures from the Gulf of Maine near shore at the top, Iceland near shore in the middle, and Iceland offshore in the bottom. And we see that the Iceland offshore fish have statistically significant narrower heads, and also statistically significant longer and narrower caudal peduncles, which is this area here in the near the fish tail. And so they do have this more streamlined body shape that's predicted by this biophysics research for reducing drag for their long distance migrations. To answer the question of how do migratory traits evolve at the genetic level, we did a genome scan. And so the basic idea here is that we group the fish into the offshore and nearshore groups. And we just look along the genome at a statistic FST, which is measuring genetic divergence between these groups. And in areas where we see allele frequency differences between the two groups, there's a peak in FST. And we do this at, the million single nucleotide polymorphisms that we have across the genome. And so when we compare the Iceland offshore and Iceland nearshore ecotypes, we can see that there's these big blocks of elevated divergence in FST that's statistically significant, more than we would expect by drift. And in the background, the yellow boxes are inversions that are in the cod genome. And so we see this elevated divergence at multiple inversions in the cod genome. When we compare the Iceland offshore fish to the Gulf of Maine nearshore fish, obviously there's a lot more less migration between these two populations. So FST across the genome is elevated more overall, but we also see elevated FST within those inversion regions. When we compare the Iceland nearshore and Gulf of Maine nearshore fish, we don't see such an extreme divergence in the inverted regions. I'm just going to see if I can hide this. Yeah, I can't find my mouse though, so I guess it's not going to happen. So inversions are areas of the genome that the DNA has essentially been flipped around and then repaired. And as some of you may be familiar, these inversions have been shown to underlie ecotype differentiation in a lot of different species for a lot of different contexts and adaptations. So for example, they've been shown to underlie color polymorphism in butterflies, size and shape variation in marine snails that are exposed to predation or not, as well as migratory shape and behavior in other species. And they've been labeled supergenes because they can be extremely large. They can be up to the size of like half the size of individual chromosomes. And there's lots of genes found within these inversions that have functions that putatively underlie these traits. But what's something that I think is really interesting is that when you look in nature, you see a lot of inversions being involved in local adaptation. You don't see just one. They also tend to be large, and reviews have shown that they also tend to be older. And so it's really interesting to think about, like, why do we see these patterns of multiple inversions involving? So this is an example from the marine snail in the middle. And each of these gray boxes in the background are inversions that are in the snail genome. And so you can see there's like 6 or 7 that are involved in this ecotype divergence in this marine snail, which is another situation where we have really high gene flow between these ecotypes. And so we know a little bit about why inversions are involved in local adaptation. When this inversion happens, it effectively reduces recombination, which in turn effectively reduces gene flow between the arrangements. And so this allows the arrangements to evolve more independently, and so divergence can evolve within these inversion regions. And we're not the first people to find the inversions involved in local adaptation or in migratory divergence in Atlantic cod. A lot of other studies have found evidence that other kinds of adaptation, like temperature or salinity adaptation in cod, as well as other migratory coastal comparisons in Norway, have involved these 4 inversions in the cod genome. And there's a lot of really interesting genes inside these inversions that could potentially have effects on migratory behavior. So for example, we see rhodopsin genes, which can mediate dim light vision. So these would be those offshore cod that are found deeper, as well as catalases that have to do with gas secretion into the swim bladder, circadian rhythm, and other genes that are found in resident migratory ecotypes and other species. So that's interesting, but all of that research doesn't really explain, like, why do we see multiple large supergenes involved in this ecotype divergence? And so a lot of the population genetic theory for inversion establishment has focused on low gene flow scenarios. So like the classical example, Kirkpatrick and Barton, in a continent island model, low gene flow evolves allele frequency differences between patches. And so in the blue patch, we see blue alleles evolve, and in the yellow patch, we see orange alleles, brown alleles evolve. And then analytical theory has shown the conditions under which an inversion has a higher establishment probability is when they're capturing these already locally adapted alleles that are weakly linked and under weak selection. But this kind of model is only looking at really low migration scenarios where you have this divergence sort of evolving due to the low migration that's already in place. And so it doesn't really explain why we see multiple large inversions or multiple inversions in these high gene flow scenarios like Atlantic cod. And so to investigate this, we developed a quantitative genetic model in a 2-patch in 2D simulation, and we used SLIM to just a basic quantitative genetic model where phenotypes were additive effects of the alleles plus some environmental variance, and each patch had a different phenotypic optimum. And we had whole genomes with like 20 linkage groups. And then we drew inversion breakpoints from a uniform distribution up to some maximum size, which was like half the size of a chromosome. And the kind of inversions that we allowed to kind of invade in these simulations were just normal inversions. So, you know, the mutation is just happening on a reference arrangement. We did not allow what I like to call intrachromosomal overlaps. And so if there was already an inversion and the simulation chose breakpoints that overlapped with that, that was not allowed. That was just computationally intractable, but it would also be biologically messy. But we did allow interchromosomal overlapping inversions. And so this would be like an inversion that happens on a reference arrangement, but it's overlapping with an inversion that had already been established in that genomic region. And so we looked at a huge parameter space. And the only time we saw inversions involved in adaptation with high gene flow scenario was when there was a really highly polygenic genetic architecture. And so we wanted to push the migration rate pretty high. So the migration rate here was 0.25. So that's like a quarter of the individuals essentially mixing between those patches every generation, which is pretty high. But when we saw inversions involved in local adaptation was also a really high quantitative trait nucleotide mutation rate with really tiny effect sizes and strong stabilizing selection. And what's interesting here, as you can see, there's some of these interchromosomal overlapping inversions on the genetic map. They're occurring in different individuals, but they're overlapping on the genetic map. So I'm going to dig more into those and explain those in a minute. So at first, I just want to break down sort of like what is happening across time in this simulation. So on the bottom is the genetic map. And each inversion is labeled according to the time that it arose. So one arose first. The first inversion was kind of drifting at a low frequency and a low effect size at the beginning of during the burn-in part of the simulation. So that's the blue star. After burn-in, we implement those divergent optimums and a period of environmental change. And then we see that very quickly inversions start out at a very small effect size, and the bubble size is the FST, and they're colored by the population that they end up having the highest frequency in. And so they start out at a low effect size, and they grow sort of like they're gaining mutations and they're gaining effects through time. And as they do so, they're also diverging. And we can look at when some of these overlapping inversions arise. And so, for example, number 7 here arises and overlaps with number 5. And what it's basically doing in this case, it's capturing divergent alleles that have already started to diverge because of the inversion is in that part of the genome. So I'm going to break this down with a picture. In this picture, we have a more inshore environment at the top, which is in our Atlantic cod, and a more offshore environment at the bottom. And at the beginning of the simulation, there's like high gene flow. And so there's some additive genetic variance that evolves during that burn-in period, but there's not any divergence between the patches yet. Then we have an inversion that happens to capture just a few weakly linked small effect alleles in a fish that has a more streamlined body shape, and that fish swims a little bit farther for food, and it has a higher fitness. That slight reduction, that inversion establishing creates a slight reduction in effective population size that allows the buildup of that inversion effect size. So that's the buildup of these brown hashes here and these blue alleles in this part of the in these individuals. and differences in allele frequency over time. So we have higher FST evolving in that area of the genome. And with this highly polygenic model, we also see a diversity of multi-locus genotypes and effect sizes within an inverted arrangement and also within the ancestral arrangement. But this is a high gene flow scenario. And so one inversion establishing is not enough to really bring the population to an equilibrium between migration, selection, and drift. And so we see the same process that's happening again in another area of the genome where an inversion captures alleles in a fish, in this case, that tended to stay near shore, that has higher fitness. It increases in frequency and effect size over time. And then we see another region of the genome now with elevated divergence. And what this theory also predicts, or this model also predicts, is that we would see both ancestral and alternate arrangements having phenotypic effects in the same direction. So this is an alternate arrangement. This is the ancestral arrangement in that patch. And the phenotypic effects in the same direction are the preponderance of blue hashes, the blue alleles. And then same thing in the other patch, but in this case, it would be the brown alleles. And then finally, after we see sort of this divergence evolving, that's when those interchromosomal overlaps tend to occur, where an inversion in the other population captures alleles under that more classical Kirkpatrick and Barton model that are already locally adapted. And then that inversion can spread in the other population. So we see multiple large supergenes involved in ecotype divergence in this model when the genetic architecture is highly polygenic and is concentrated inside inversions. So now we can go back to the data and see if some of the empirical patterns kind of match up with these theoretical patterns or modeling that come out in the modeling. So we're going to dig more deeply now into these individual arrangements and how they come together within individual fish. And so the reference that we align these data to was an offshore cod in Norway, so putatively migratory cod in Norway. And so if we saw migration the same way in Iceland, we would see all blue for these arrangements for the offshore Iceland fish, but that's not what we see. The offshore Iceland fish tend to have alternate arrangements on the inversion on the first and the seventh chromosomes. And so it raises this possibility that the migratory basis or the genetic basis for migratory behavior in Iceland could be different than Norway, but we would need more Norwegian samples to really test that. We can then compare that to the nearshore fish, and the nearshore fish tend to have alternate arrangements on the second, and the inversions on the second and the 12th chromosomes. And the other thing that's really interesting that I want you to take home is that each row in this heat map is an individual. And so if you look across at any one point, you can see that these arrangements are coming together in a variety of different ways within an individual. So now let's look for some of the patterns that we saw in the simulations with this highly polygenic scenario. First question that I'd like to ask with the data is there evidence for diversity of multi-locus genotypes spanning the region affected by the inversion? In the simulations, we didn't see that every arrangement had exactly the same alleles. There was definitely a lot of diversity within the arrangements. So now I'm going to zoom in to the single nucleotide polymorphism genotypes for that inversion on the first chromosome. And we see an incredible amount of diversity within the inversion, to the extent that almost every fish has a different multi-locus genotype. But you can also see, that inversion, that alternate arrangement here by the preponderance of alternate SNPs in those offshore Iceland cod. The next question we'd like to ask is, how are these different arrangements associated with the morphological traits that we measured? And do they tend to behave as a single locus with large effects on these traits, or a highly polygenic locus with a continuum of effect sizes? And it wasn't immediately clear like how to answer this question. We have a lot of traits that are affected by the genome. We have multiple inversion arrangements within the genome that are coming together in different ways within individuals. And we have unique multi-locus genotypes within the arrangements. And I like to call this the matryoshka or the Russian doll problem. It's like, you know, we have layers within layers within layers of our data, and we're trying to figure out how everything kind of fits together. And so what we needed was a method that predicts a multivariate trait value from a multi-locus inversion genotype. And you might think I'm going to use some kind of fancy AI to do this, but I asked AI to generate some fish Russian dolls for my talk, and it generated these weird like bird-looking fish things. I don't know what those are. So obviously this was a sign AI was not going to work. We can just use the tried and true statistical methods that have been developed for multivariate analysis. And recently I showed that a redundancy analysis, which is essentially a way to associate multivariate environment or multivariate trait data with multivariate genetic data in a quantitative genetic model, these individuals will map into RDA space according to the sum of the additive effects of their QTNs or their quantitative trait nucleotides on a trait, even if none of those quantitative trait nucleotides are outliers in the traditional sense of a genome scan. So for example here, for these measurements in the tail, this group of offshore Iceland fish are mapping sort of like along this axis, which means that they would have longer values predicted from this constrained ordination on their genotypes and phenotypes. So we can run a local redundancy analysis on each inversion to get an idea of how these individual inversions are associating with multivariate traits. But this is hard to interpret. There's like a lot of cognitive load involved in trying to understand how everything's mapping in multivariate space here. And in this study, I also showed that a simple back transformation of RDA space can accurately predict a standardized trait value, or like a Z-score, for each individual based on their multi-locus genotype. And this Z-score is basically the product of the loading of the individual on the first RDA axis times the loading of that trait value on that RDA axis times the proportion of variation that RDA axis explains, summed over all RDA axes. And I'm just going to digress for a minute to show you that I evaluated this RDA back transformation on a bunch of simulations, including this 6-trait multivariate adaptation scenario. And so in this scenario, that we simulated in SLIM, there's 6 environments that is coming from real patterns of environmental variation that we downloaded from British Columbia. And individuals are having to adapt to all 6 environments at once. And there's a burn-in period, and after the burn-in, there's like a few refugia at the bottom of the landscape. I don't know if you can see this, but all these little dots are individuals. And I'm just going to fast forward it here. So, oh, I guess it didn't like that video. But what you could see is that it is the population kind of expands across the landscape in the video, and it looks really cool. So if you want to see it later, I'll show you on my laptop. And so in this simulation, there's like these complex patterns of admixture that evolve on the landscape. So there's some considerable structure. And I evaluate, and another interesting thing that happened in these simulations is that this, again, these are highly polygenic traits adapting to multiple environments at once. But we didn't see the traditional sort of allele frequency climb patterns that we might expect with environmental adaptation. We saw a lot of these non-monotonic patterns in allele frequencies. But nevertheless, this RDA trait prediction was still accurate in this scenario because individuals map according to the additive effects of their quantitative trait nucleotides, which is still a linear relationship. So we compared the accuracy of the RDA predicted trait value to other methods, including a polygenic score based on outliers from genetic environment associations. So the y-axis here is basically a measure of accuracy. It's the correlation between the prediction and the simulated phenotype. And the x-axis is each of these different environments which had like an environmental trait that it was adapting to. And so in some situations, in some environments, there would be low accuracy with genetic environment association outliers, either because there wasn't power to detect them or it had a high false discovery rate. Or in other situations, we would see low accuracy because of structure correction. Another thing this paper showed is there's not really a straightforward way to correct for structure when you're in these like multivariate environmental landscapes. But nevertheless, this RDA back transformation could accurately predict the standardized trait value as a z-score in all these different scenarios, even when there was some nuisance, like nuisance environmental variables that didn't have, they weren't involved in selection on the landscape. So now we can use this RDA trait prediction to answer some of these questions. So first, within an ecotype, do inverted regions have phenotypic effects in the same direction? So you may remember the model showed that ancestral or in alternate arrangements had phenotypic effects in the same direction. And to show you that visually, on the x-axis here is that standardized trait prediction from the RDA based on an individual's multi-locus inversion genotype. And values to the left are essentially shorter predictions for shorter trait values, and values to the right are predictions for relatively longer trait values. And each color is an inversion. And so we can see for the Icelandic offshore fish, the multi-locus inversion genotypes that they have tend to predict shorter head values. So they had much narrower heads, whereas the Gulf of Maine nearshore fish tended to have much wider heads, and the Icelandic nearshore fish were either like the Gulf of Maine nearshore fish or somewhere in the middle between the two. And just as a reminder, the Icelandic offshore fish had the alternate arrangement on the first linkage group, the reference on the second, the alternate on the seventh, and the reference on the 12th. So no matter which arrangement they had, the arrangement had phenotypic direct effects in the same direction, which I think is really interesting. And then the opposite was true of the Gulf of Maine nearshore fish and the Icelandic nearshore fish. We can do the same kind of analysis with the traits that are in the caudal peduncle or near the fish tail. And we see that the Icelandic offshore fish have multi-locus inversion genotypes that predict longer caudal peduncles and shorter caudal peduncles, whereas we see the opposite in the, or narrower caudal peduncles, in the Icelandic offshore fish, and we see the opposite in the Gulf of Maine nearshore fish. And again, for the Icelandic nearshore fish, we see patterns that are like the Gulf of Maine nearshore fish or somewhere in the middle. And so that is also a pattern that is consistent with what we saw in the simulations. So now we can test, well, do we see a diversity of effect sizes within the ancestral arrangement? In these simulations, there was like an average effect size for an inversion, but there was some variability around that because of this highly polygenic architecture. And so do we see that in the data? So again, we can use that standardized trait prediction from the RDA to look at the distribution of putative effect sizes, at least as a standardized z-score, and ask, do the inversions behave as a single locus with large effects or as a highly polygenic locus with a continuum of effect sizes? And we see these kind of multimodal distributions. It's pretty obvious on the first linkage group where the three modes correspond to the three arrangements that are segregating in the population, but we see a variety of effect sizes around those nodes. And in other linkage groups, kind of see that pattern, but it's a little bit more continuous. So we kind of see both. We see these multimodal distributions that are consistent with polygenic effects on top of these larger effects of the supergene arrangement. And then finally, we can ask, do we see evidence for overlapping arrangements on the genetic map in different individuals? And as a reminder, we're talking about these interchromosomal overlaps. These are inversions that are in different individuals that are segregating in different populations, but they kind of like overlap on the genetic map. And so the population genetic signal you can't really see them in these FST peaks because everything's already diverged. And so to really look at this, we're starting to call structural variants and look for exact breakpoints in the genomes. And so my student right now is running these programs called DELI and GRIDS to detect structural variants in our data. And this is just an example of some of the output. We're just starting to think about like how can we kind of test this modeling prediction. But to understand this output, each cluster of colors are the inversions found that are being called by these programs within an individual. And each row is just like a different call. So we've got one here, one here, one here. They're all small. Some of the programs are like, picking up on the same thing twice, or two programs are finding, the same thing. And so we have to we're still kind of figuring out a way to like collapse these down into like what we're going to call an inversion and what's not. And I'm thinking about long reads to verify some of this stuff. So there's a lot of work to do. But what I think is interesting is that if we take, for those of you that are familiar, you can use PCAs to kind of look at where the inversions are in the genome. And so what I've highlighted here in yellow is the location of the inversion that we estimate from the PCA population genetic data. And it does kind of look like we have a lot of potential inversion segregating in that area in the genome. So I'm really excited to kind of test this modeling prediction. I don't think anybody's really done this before, and it's, I think, a cool new direction for research. So to summarize, we see more streamlined body shape in offshore cod, and genetic differences between offshore and nearshore cod that correspond to inversions in the cod genome. We see multiple large inversions involved in ecotype divergence under high gene flow, like is happening in cod, when the genetic architecture is highly polygenic and it becomes concentrated in these inversions. And then finally, we see evidence of empirical patterns that support theoretical predictions, such as all of the arrangements within subpopulation or DEEM are kind of like having phenotypic effects in the same direction. We see a lot of diversity within these inversions in terms of genotype diversity, as well as effect size distributions consistent with large effects of inversions with polygenic effects on top of that. But still begs the question, these are pretty subtle phenotypic differences between the fish, right? So could these differences in fish shape matter for fitness? And so to try to answer this question, I dug back into the biophysics drag literature. And I found a couple of studies that are really interesting. So in this study, they looked at the amount of resistance that was caused by different parts of the fish body. So they looked at the head and then these three areas of the midsection of the body. They didn't look at the tail. And what they found was as flow velocity increased, by far the head causes the most resistance on the fish when it's swimming. And then they compared two species, and they didn't put their axes on the same scale. So I kind of like stretched this out so you could see them on the same scale. But the second species, which was much less streamlined, had even more resistance in its head shape. So that's an interesting study, but it is on different species. So it's not quite as powerful as the second study that looked at more subtle differences in fish state within a species. And so on the x-axis in this graph, the deeper bodied morphometric fish by morphometrics are on the left side, and the more streamlined bodies are on the right side. And the arrows show the speed of water, and then the fish that are remaining as the water speed increases are shown in the graph. And so as the water speed increased, only the streamlined fish were able to maintain swimming at the higher water speeds. And this paper specifically said a narrow caudal peduncle was more efficient for maintaining swimming speed, which in turn would turn into fitness in these Atlantic cod migratory genotypes. So I think it's really interesting that the evidence we found in the biophysics literature lined up pretty well with what we're seeing in the field.
But what's the cost of it?
What's the cost of the streamlined body shape? Like, why aren't all fish streamlined? Yeah, that's a good question. I don't know. There must be some costs associated with it, right? Because otherwise you would see more streamlined. I mean, in general, most of the cod are pretty streamlined, and the differences we're seeing are pretty subtle. So in summary, 4 highly diverse inversion regions with polygenic effects on morphology act together on a migratory or less migratory body shape in Atlantic cod. And this is evidence in favor of a highly polygenic basis for inversion of evolution under hygiene. So with that, I would like to thank you for your attention and thank you for the invitation, and I'd be happy to take your questions.